त्याची लेअर जास्त आहे. हॅलो 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 हॅलो
Okay, so once again, thank you all for coming. And this we are now towards the last lecture of the series, and it has been a very nice day outside. At least not the night, maybe it was raining a lot, but outside it's so nice, green, and this whole campus is very beautiful, is what I would say. Very close to nature, which is not what you usually see in universities because mostly they are buildings, buildings, buildings. So it's really nice that way. So now we move to an area of um, basically we have been talking about immunity. It's not that the immunity in mucosa is very different, but as I have been mentioning, the whole of the mucosa, you are exposed to many different things. Your skin is also a mucosal organ, nasal passages, gut, reproductive areas. All these are more exposed to uh, both harmful and pathogenic things. So the mucosal immune system has evolved in such a way that you respond, you try to minimize response to harmless things and respond to something which is harmful. And this mucosal immunity is become is also something which has become more and more important in the last 10, 12 years when people realize that the systemic immune response which you see in the blood and the spleen may not be same as what you would see in a lung or in a gut. There is a difference. So if you're devising therapies or if you are looking for some infections in that area, you need to understand the immune response in those areas first. माउस निकाल. Okay. So what is common in the whole of mucosa is that all these are barrier organs, right? So so basically some people also call it barrier immunity because of that. And barrier immunity, basically if you see in the skin and the mucosa, in all these cases you have the epithelium lining. So, and that forms a barrier, so it is barrier immunity. And different names have been given, like mucosal tissue associated immune response. So some people call it MALT. Now you will see that there are 110 acronyms of this kind. For nasal it is separate, for gut it is separate, for lung it is separate. So these are all simply different ways of referring to immune response in different areas. And I would not worry too much about it these days. In most of these mucosal areas, is what you will find is that there are microbes, which commensal microbes, which live in these areas, and that's why they are known as commensal. And they play an important role. So homeostasis is maintained by a combination of mechanisms that inhibit inflammation and promote tolerance. So that is what I've been saying, that in the mucosa, the immune system exists in a slightly suppressed state. It's not tolerized to the state like, okay, that it cannot respond to anything. But if you compare the spleen and the, say, gut or cells, there is a difference. There is more T regulatory cells, there is more of less of activation. But still it's not that we are getting infected every day through these areas, because side by side surveillance or other things are increased to compensate for this immunosuppression. So this is just to show you which different areas are there. So you start with the oral, then this is airway, this is intestinal, this is skin, and this is the cervical part. So this is all what constitutes the mucosal immune areas or barrier immunity areas. And maximum work has been done in this intestine, and I would say lung, but more in the intestine than in the lung, because intestine is the largest of organs. Even though skin, a lot of work has been done, because skin is the largest organ, if you see, which is exposed to the outside. So this is basically reiterating what I said, that all barrier surfaces are lined by epithelial cells, and epithelial cells, it's not like they're stationary cells and just forming a barrier. They actively participate in the immune response. Like if you remember the toll-like receptor and PRR receptor lecture, I told you that epithelial cells also express this toll-like receptor, so they can sense, they can do immune surveillance, they can sense if there is any infection coming in. And many times they are also the first cells to respond, because these are the cells which are in contact with the outside. So when they respond, they produce some cytokines and all, and sometimes they are the ones which will activate the immune system. Side by side, I'll show you later that cells like dendritic cells and all can project out their dendrites in between the epithelial cells and use them for sensing, like a periscope in a this submarine. So some produce mucus within the epithelial cells. Usually you have cells which will produce mucus, and they also secrete a number of antimicrobial peptides, the epithelial cells, which, because by the time immune system is getting activated, in any case all cells are trying to contain whatever is coming in. That is the bottom line. So it's not that other cells will just stay there and do nothing. If you see from an army point of view also, the front line which is there, they are trying to shoot or do something or the other to keep the invaders in check while they get more reinforcements, right? So it is like that in this case also. Then they also have cilia, especially in your nasal passages, which helps sweep the pathogens away. And when the epithelial cells become damaged, either their adherence goes away or if they become infected, they also secrete a number of things which would alert other cells that there is a microbial invasion. So barrier organs are populated by both innate and adaptive immune cells, and mostly very near the barrier, it is mostly the innate immune cells, because innate immune cells are the ones which will start the immune response. So, and adaptive immune cells are usually found in secondary lymphoid organs, right? So, in this case also you have things like lymph nodes, and also some areas in different, which I will show you, which are specific pockets of which behave like lymph nodes, and these are specific pockets of B and T cells, especially in the intestine. So these are all different kind of cells which are present, and these are, you know, the common immune cells which we have been talking about, except maybe this innate lymphoid cells. Innate lymphoid cells are basically cells which behave very much like a T cell, but they don't require any antigen presentation. The difference is they secrete cytokines which are similar to what T cells would secrete and not the macrophages and other cells, but they don't require, as I said, antigen presentation. And most of the innate lymphoid cells seem to be present more in the mucosal regions than outside of it. And this is again a relatively new cell to be discovered compared to other cells, and this is even rarer than dendritic cells, so it's harder to study. So and innate immune lymphoid cells also, you know, like I told you, the T cells come in, T helper cells come in different flavors like Th1, Th2, Th17, Tregs. Innate lymphoid cells also come in this ILC1, ILC2, ILC3, same kind of classification. So local lymph nodes, as I mentioned, one of them is mesenteric lymph node, and these are especially associated with the intestine. And these are what I told you, that small pockets. These are known as Peyer's patches, because you have all these epithelial cells, and you don't want the lymph node to be here for those. It's too far. It may take a long time. So you have inside only some pockets where the DCs and macrophages or whatever the innate immune cells, when they get activated, they can go there and activate the T cells. They don't have to go too far. So this is what I was mentioning, that different names are given. When you have a nasal associated lymphoid tissue, then people call it NALT. So skin associated SALT. So all these are acronyms which people use. So ALT is associated lymphoid tissue, and you can take the first letter from whichever organ you are studying. This is just showing you that there is, this is the epithelium. Especially in case of intestine, you have these villi, right? And in these you have small pockets like this. I have a video which I'll show you a little later, which it is a little long, in the sense 4 or 5 minutes, but I think the reason I like that video is it shows very nicely whatever I mentioned. Then I think it is going to be much easier for you to understand. So this is exactly what I've been saying, that it can barrier immune systems initiate tolerogenic and inflammatory response to microorganism. And at homeostatic, basically at the baseline, when there is no infection, it is tolerogenic, because you have so many commensal bacteria and others there. You don't want to make an immune response against them. That's why you have a tolerogenic kind of. But one thing I need to mention, so there are different levels of tolerance, okay, like suppression. So in this case, the immune system is suppressed, but not to the extent you would find in a tumor microenvironment, or where, so it's like different thresholds of suppression. Because if it is through that level, you have seen how difficult it is to break that tolerance. So you don't want your immune system to be suppressed to such an extent that you need some outside therapy for it to become activated. So this is suppression, but here, compared to the, what is there in cancer. Because what is happening, if it is suppressed to slightly, then what happens is, as I said, it does not react to commensals, but it can immediately react to any kind of pathogens which come. So, and this tolerance in the mucosa is an active suppression. Like, it is not simply that, okay, it is suppressed. There is cell-to-cell interaction and cell-microbe interactions. So basically, epithelial cells, it is known that they produce molecules like this TGF, this is wrong, TGF beta and IL-10, which induce T regulatory cells. So even without microbes, epithelial cells in general secrete TGF beta on GM-CSF. And side by side, these commensal microbes which are there, they can interact with the immune system. Especially, I can tell you about dendritic cells, that when they interact with the dendritic cells, we don't get upregulation of co-stimulatory molecules or MHC molecules. In that case, when these dendritic cells activate T cells, antigen is presented, but the second signal is not provided because co-stimulatory molecules are not going up. So then you get formation of T regulatory cells or T suppressor cells. So remember, in the tumor, a similar thing is happening, that in that case, either there is no co-stimulation or you have inhibitory co-stimulation. So that's why the Tregs were increasing. But here, it's mostly that Tregs are being induced. And I already mentioned about that they secrete a number of antimicrobial peptides. Some of them you may have heard about, like defensins and beta defensins, alpha defensins. All these are produced by. So this is just showing you that these are microorganisms. You are getting tolerogenic DCs which are making IL-10, and you're getting these T regulatory cells. And these T regulatory cells also produce IL-10 and TGF beta. So this is suppressive, this is suppressive. So that is why the whole atmosphere becomes suppressive. And side by side, other things can be happening if there's an infection. you have an activated DCs, and all these are ILCs, are innate lymphoid cells, and you have activated T cells, the Th1 cells making interferon gamma, the Th17 cells making IL-17, and then if uh, these cytokines are being made and DCs are becoming activated, it would suggest that there is an infection. So you have attraction, other cells also now coming into the area, because this is kind of an alert for other cells, immune cells, to come in. In fact, IL-17 is known to actively recruit neutrophils. It is a chemoattractant for neutrophils along with IL-8. So it attracts all these cells inside so that they can fight infections. So this is what I was mentioning, that it is known that dendritic cells can extend their dendrites like this in between the epithelial cells to survey for the any pathogenic or even commensal microbes. And this has been shown in the intestine in an experiment.
Hello, ma'am. Ma'am,
Yeah.
why some people they always make more mucus when they wake up in the morning? Why they always have excess of mucus production?
In the morning, but I think it's mostly because it is dysregulated in those patients, because it is not a common thing that all of us have increased mucus production. You will see mostly it is with age, or because you have either suffered from some infection, maybe remnants of that infection remain, and that leads to more of the mucus production. And when you are moving about, it is cleared more easily. When you're lying down, it doesn't drain as easily into your and removed. So that is why I think many people suffer from this congestion in the morning. Yeah.
If there is an infection, or they will help to regulate it. But then why regional group of lymph nodes they get activated then?
Because
MALT is also a lymph node, so there the activation has started.
so basically the DCs will migrate to the nearest lymph node also, and even in the intestine you have these patches, Pierce patches, right?
Yeah.
But if you're
Anywhere
talking about
mucosa for that matter.
yeah, but mostly these are concentrated pockets, the lymph nodes. So that is why the DCs will migrate there and activate. So in oral and in lung, it is known that they migrate to the areas which like these. Yeah. So that's why they go there, and that's why if the cells are multiplying, you have this swelling of the lymph nodes, because now T cells are proliferating. So
Ma'am,
that's,
cancer cells can exploit this mechanism, no, ma'am. Suppose some mucosal tumors are there, right? So it can produce more mucus so that it can encapsulate inside, so that immune system cannot touch.
definitely, because MUC1, mucin genes are known to be mutated in many cancers. They are overexpressed in many cancers. So that maybe that is one
In
of the.
that case, immune system will not act. Even chemotherapy will not penetrate inside if mucus is covered.
If it is very very thick, then only. If mucus is very thick, then penetration is difficult. But maybe you can couple it to some kind of an enzyme which will dissolve the mucus, and then the therapy can go in. But generally in tumors, mucus, I have not heard of such thing that there's so much of mucus that the therapy cannot go in. I haven't seen.
With respect to ovarian cancer, it is making huge amount of CA125. CA125.
Okay.
So they produce a lot, ma'am. 35 unit only normal, but it will go up to 10,000 unit per ml.
So it can go up to that level, but generally to form a thick mucus, the clearance also of the mucus also has to be defective, so that it kind of gets deposited there. So that is one of the mechanisms where cancer cells will employ, but generally it is, I have not seen, okay, it's amongst the many mechanisms that the cancer cell employs to suppress the immune system. But only that is never a problem. Okay. So now if you come to intestinal immunity, you have all these different organs. So you have the stomach, and in the stomach you know that the pH is very very acidic. So you do have lactobacilli, but if you see the number of microbial in the stomach is much lower. It's 10 to like 100 to 1000 microbes. Okay, this is your jejunum. Here it increases 10 to 5, less than 10 to 5. In this ileum, you have 10 to 3 to 10 to 7. What I'm trying to say, your large intestine is the area where you find the maximum number of microbes. And if you see, the diversity is also increasing. So many different species are present. Mostly in the upper part, you find mostly lactobacilli and streptococci. So this is just to show you how the gut is organized. And now a lot of sequencing is being done, and people are thinking that Firmicutes is better than Bifido or whatever it is, but still I think it is a lot more needs to be done before we can really understand what is happening. So for this, I think I will leave this part in the sense I can tell you, but I think you will understand it more from the video because it is like a cartoon. So basically, it's telling you the different kind of cells which are there. So the intestine has these villi, right? And in the crypt, you have these Paneth cells, which make different things. And one thing which I need to mention is that the B cells, the antibody which they make primarily in the mucosa is they do make IgG, but IgA is the primary antibody which is made. It seems to be more effective against mucosal pathogens. And there is active transport of, and these M cells which are there all make mucus and they help in transport of antigens, basically. And there is an outer mucus layer and inner mucus layer. So as I said, I'm going to show you all this in that video. I will come to that straight away, I think. So this is simply, you have the mucosa. This is the area in where you have IgA producing B cells and plasma cells. And then you have the lumen and the submucosa. Okay, so you have different type of epithelial cells. I think I'll just go straight to that video first, and then we will come back because I think it will be like a repeat of what I'm trying to say. So here, the only thing I can come back to this. First you look at that, and then I will come back to this. Here. I think it is not connected to the internet. Okay, then I think we haven't logged in. That's the only, you can go ahead and log in. So, but meanwhile, you can log in. By that time, I'll go back and talk about some of the other things. Oh, it won't move with this. I got confused. लॉग इन करना पड़ेगा. खुलेगा खुलेगा, इसमें थोड़ा आराम से होता है. Because this videos for the mucosal immunity for different organs, these are made by Nature group, Nature journal group. Even though they are slightly, I would say, older in the sense they are from the year 2013 or 14. But when I was looking at it and I've looked for many videos, it kind of tells you everything very nicely. So, and it shows you also. So I think it's easier to remember in that case. That's why I was saying. उसमें हाँ बस क्लिक करेंगे तो आ जाता है साथ में। हाँ वाईफाई कनेक्ट करना पड़ेगा ना, वही मैं बोल रही थी, लेकिन कनेक्ट कर देते हैं क्योंकि इसमें जो है वो लंग वाले में भी है फिर। अभी 2 मिनट में वो पूछेगा। हाँ हाँ, पहले वो आ जाने दीजिए, लॉगिन हो जाएगा तो उससे क्लिक करने पे ही हो जाता है फिर सीधे। GIAN वाला यूज कर लीजिए। ठीक है ठीक है, कोई भी करिए, कोई फर्क नहीं पड़ता। अब वापस से हाँ ओके, अब पहले उस पर पहुंच जाते हैं स्लाइड पे। It won't click till you put it in the slide, show mode. हाँ, क्यों वो पता नहीं। अब क्लिक करें, आएगा आएगा। मुझको भी अब अकल आ गई कि इसी में it takes time, this one. I thought okay. Patience. Just keep like ठीक है, हो जाएगा फिर उसके, है ना, एक बार लोड हो जाएगा तो हो जाएगा। फिर तो हम क्लिक कर देंगे, थोड़ा बड़ा है ना इसलिए टाइम लग रहा है लोड। ओके
The human gut can be the scene for devastating conditions such as inflammatory bowel disease, which arises through an improperly controlled immune response. The gut is often the body's first point of contact with microbes. Every mouthful of food is accompanied by a cargo of microorganisms that go on to encounter the mucosa, the innermost layer of the gut. Most microbes are destroyed by the harsh acidic environment in the stomach, but a hardy few make it through to the intestine. The intestinal surface is covered with finger-like protrusions called villi, whose primary function is the absorption of nutrients. However, these structures and the underlying tissues also host the body's largest population of immune cells. Scattered along the intestinal mucosa are dome-like structures called Peyer's patches. These are enriched in lymphoid tissue, making them key sites for coordinating immune responses to pathogens whilst promoting tolerance to harmless microbes and food. The villi contain a network of blood vessels to transport nutrients from food to the rest of the body. Lymphatics from both the Peyer's patches and villi drain into the mesenteric lymph node. Within the villi is a network of loose connective tissue called the lamina propria, and at the base of the villi are the crypts, which host stem cells that replenish the epithelium. Finally, the epithelium, together with its thick overlying mucus, forms an important barrier against microbial invasion. Embedded within the matrix of the Peyer's patch is a mix of immune cells including T and B lymphocytes, macrophages, and dendritic cells. A key function of the Peyer's patch is the sampling of antigens, in this case mostly bacteria and bits of food. To facilitate this, the Peyer's patch has a much thinner mucus layer as well as specialized phagocytic cells called M cells, which can transport material across the epithelial barrier via a process called transcytosis. Finally, dendritic cells are able to extend dendrites between epithelial cells to sample antigens that are then broken down and used for presenting to lymphocytes. Sampling antigens in this way typically results in so-called tolerogenic activation, where the immune system initiates an anti-inflammatory response. With their cargo of antigen, these dendritic cells then traffic to the T cell zones of the Peyer's patch. Upon encounter with specific T cells, the dendritic cells convert them into an immunomodulatory cell called a regulatory T cell, or Treg. Defects in the function of these cells are associated with inflammatory bowel disease in both animals and humans. These Tregs then migrate to the lamina propria of the villi via the lymphatics. Here, the Tregs secrete a molecule called IL-10, which exerts a suppressive action on immune cells within the lamina propria and upon the epithelial layer itself. IL-10 is therefore critical in maintaining immune quiescence and preventing unnecessary inflammation. However, a breakdown in this process of immune homeostasis results in gut pathology, and when this occurs over a prolonged period and in an uncontrolled manner, it can lead to inflammatory bowel disease. Chemical, mechanical, or pathogen-triggered barrier disruption, coupled with particular genetic susceptibilities, may all combine to set off inflammation. Epithelium coming into contact with bacteria is activated, leading to bacterial influx. Alarm molecules released by the epithelium activate immune cells, and Tregs in the vicinity scale down their IL-10 secretion to enable an immune response to proceed. Dendritic cells are also activated by this environment and start to release key inflammatory molecules such as IL-6, IL-12, and IL-23. Effector T cells also appear on the scene, and these coordinate an escalation of the immune response by secreting their own inflammatory molecules, tumor necrosis factor alpha, interferon gamma, and IL-17. Soon after the effector T cells have arrived, a voracious phagocyte called a neutrophil is recruited. Neutrophils are critical for the clearance of bacteria. One weapon in the neutrophil armory is the ability to undergo a dramatic form of self-destruction called netosis. This leaves behind a jumble of DNA saturated with enzymes called a neutrophil extracellular trap. Although this can effectively destroy bacterial invaders and plug any breaches in the epithelial wall, it also causes collateral damage to tissues. Slowly the tide begins to turn, and the bacterial invasion is repulsed. Any remaining neutrophils die off via apoptosis, a non-inflammatory form of cell death, and are cleared by macrophages. Epithelial integrity is restored by replacement of any damaged cells with new ones from the intestinal crypts. Finally, Tregs are recruited once again to calm the immune response. Targeting the molecules involved in gut pathology is leading to effective therapies for inflammatory bowel disease.
Exit. So the reason I wanted to show you this video is, in this, all the different cells types I've been mentioning, it tells you how they will function at homeostasis. And so you have these M cells which can transport antigens from the lumen to the inside. you have the dendritic cells which can sense antigen from the outside to the inside. Then also you have so in normal conditions, I need to shut down. So you also have stem cells there in the crypt, which can, renew the epithelium then and there. You don't need some traveling from far off, cells coming from the.
Ma'am, what exactly happening, ma'am? Transition. You are saying that a minimal level immunity is there at the lumen. That
Suppression, right?
yeah. But how the transition is happening for IBS patient?
So IBS patient, for whatever reason, as you can see, it depends if there's a major infection or maybe there's an injury because of something. It could also be some kind of a spice, I don't know. So it leads to rupture of the epithelium. Maybe not rupture, but some epithelium is no longer as intact as it was before.
Permeabilization is more.
Yeah, permeabilization is more. So then you have all these bacteria can enter. And that causes.
So if we seal the permeabilization, then we can.
Yes, but so seal the permeabilization, the way you can seal is that there should be new epithelium being made, which should function normally, right? So the problem is
infection only causing the permeabilization.
Yes, because when epithelial cells become activated by any bacteria, the adherence goes down.
The tight junction protein will be downregulated.
It is very well established, and also the inflammatory cytokines which are secreted by the immune system cells also alters the.
So if pathogen if it is not going inside the membrane, then no issue.
No issue. There's always, usually it is believed that this inflammatory bowel disease, it starts with some problem in some injury or something in the epithelium. Yeah. So that now the problem is the immune system no longer remains tolerized. So it is, and it starts to react more and more.
What is the treatment for IBS, ma'am? Because my friend is taking daily one tablet. I don't know what he's eating.
So IBS, I haven't, I think it is simply, there's no, it is a long-term maintenance treatment, okay?
Yeah.
So the tablet
there is no cure for it.
There's no cure for it. Right now people are thinking about a little bit of immunotherapy, but it's still, I would not say that.
Because if we keep taking anti-inflammatory for a long time, then we may develop TB.
It's a lot of, so it is a lot of problem. I totally agree. But I haven't seen a very clear, I see people in long-term maintenance only, and I think they have these episodes where it flares up more, and then there are times when it is more quiet, but doesn't go away as such. And that is why people are trying to look at the immune response, and especially now I think these IL-17 producing T cells are supposed to be a major.
Surgery help, ma'am? Surgery?
No, not really. Because if it is only a small area, because surgery you can't really remove the whole of the intestine, right? And in this case, unless it is very localized, you can remove part of it. Otherwise, how do you localize? Like it may have spread in different areas.
So it means if we have not understood anything, how that is.
No, that is one. So this is not my recommendation, but I think increasing curcumin and some of the anti-inflammatory foods in your diet may help in some ways, because it is very well known. We know from a long time that this turmeric, and the active compound in turmeric is curcumin, and curcumin is known to be an anti-inflammatory. It is also known for internal healing, for that matter.
But some report says curcumin is reducing immune response.
No, so if you're going to drink it by gallons, then maybe it will affect the immune response. Otherwise, you eat it, it is cleared up by your system. It doesn't go there and accumulate or something like that, that it is going to suppress. Otherwise, all of us should have highly suppressed immune system, right? We are having it morning, evening, night. Like we have it in all, foods. So
no, the thing is, if it is the IBS is because of pathogen only, then why particularly that area? Why not other places? Like why only a bowel?
Because the injury is in the bowel. That is why other areas will result in other kind of inflammatory diseases. In the lung, you have asthma and other problems, right? And so similar. It's simply that wherever it is located, the particular. So it's a kind of an autoimmune disease. So if it is in the brain, multiple sclerosis also displays a similar kind of symptoms, but it won't affect the bowel. It affects the brain. Th17 cells are also involved there. So in all cases, what you see is an immune inflammation, loss of T regulatory cells. So that is one thing which is very, very common amongst many autoimmune diseases. That's why if you see, I have told you that a lot of people are trying to develop methods where they can generate large number of Tregs and they want to infuse back into patients. So that is definitely going on. So now I can go back and talk about these things if you want, but I think most of it has been kind of shown, but I'll go very fast with all these things, okay? We'll start from here. So epithelial cells, as I told you, also express toll-like receptors, and generally at homeostasis, since they are interacting with the commensals, it does not result in an immune response. Instead, it leads to generation of Tregs. That's what you saw in the video also, in the first part, when there is a intact epithelium. Okay. Epithelial cells and antigen-producing cells also produce. These are two B cell activating factor and APRIL, which basically can produce IgA synthesis. Because IgA is very good at neutralizing bacteria, and I will come to that later also. And I think I've already shown you that when there are so many cells which produce IL-10 in the intestine. This is going into more specific subsets of dendritic cells. So generally it is shown that in the mucosa, the dendritic cells express a molecule known as CD103, which kind of allows them to remain in that area. It makes them interact better with the epithelium. So it remains there, otherwise they may go along with the circulation. So many of the dendritic cells, so in all organs, something is there to retain the cells so that they remain there, and when the infection comes, this adherence goes away and they can move. So this is one of the molecules. So this is just showing that so many different things can be produced by the epithelium, and this is activation via TLR5. TLR5 usually recognizes the flagella on the bacteria. So that is why the TLR5 expression is much higher in the intestine, because most of the bacteria which infect the intestine have the flagella. Not all bacteria have, but that's why it is more expressed in the. So this is just showing you different kind of cells, the CXCR. So I would not worry about these receptors and things unless you are studying this in great detail. Then you need to understand that, okay, what is the CXCR3 R1 kind of macrophage? What is the macrophage which does not express all this thing? But it's what the major thing which I should emphasize is that the immune cells are similar, but they seem to have slightly different expression of some markers which make them more functional in that particular area. And like B cells are producing more IgA than IgG. So there are some differences. So this is just to show you how many different molecules are produced and what is their function. So I had already mentioned these three molecules many times, and this RA is basically retinoic acid. So it has been shown very clearly that epithelial cells produce retinoic acid, TGF beta. So they act on dendritic cells to induce tolerance, apart from the commensal bacteria also inducing tolerance. So many mechanisms are there to keep the immune system suppressed in the intestine, in the mucosites, what else. And these are basically B cell factors, so they will enhance IgA production. IgA itself is a little bit more tolerogenic than IgG. So that's also one of the functions. These include AMPs is simply antimicrobial peptides. TSLP is thymic stromal protein, and basically this is also produced by epithelial cells. It is also known to suppress. So that this is just telling you that how many different molecules are produced to keep the immune system in check. But these, now we are coming to molecules which are produced in response to infections, and they can be tolerogenic. So generally, these ones are not tolerogenic, even though it is written, and they usually induce um, an immune response. The only one I would say is IL-22 is not really tolerogenic, but IL-22 is a cytokine which is secreted by our T cells and also innate lymphoid cells, but it does not have a receptor anywhere else. There is no receptor anywhere else in the body except in on the mucosal epithelial cells, and it helps in mucosal repair and secretion of repair and regeneration. So if there is an infection, it has infection goes away. Now everything has to go back to normal. So whatever cells were made, too many of them, they die, but also the epithelium is injured when there is an infection. So from the crypt, you get new cells being formed, but IL-22 enhances this regeneration repair. So if you have a defect in IL-22 production, these epithelial cells are not going to repair very well.
Ma'am,
Yeah.
I have read somewhere JV virus is there, ma'am, Japanese encephalitis virus, that is going inside the brain, right?
Yes.
So it what it does, it seems it is blood-brain barrier. It is
it breaks the blood-brain.
So basically, it is generating VEGF production and down-regulating the tight junction proteins. That is how it enters. That is the hypothetical.
Yeah.
So same way, can we make a therapy to? Since you said that the leakage is happening, the pathogen is going inside and causing the inflammatory disease. So can we make a target? So we should find a protein which is responsible for the.
So you want to enhance the adherent, the tight junction, right?
Yes.
So
is it possible?
Basically, then you want this to enhance, right? IL-22 or some other mechanism which should enhance the repair of the epithelium. That's what happens in the repair, that you get rid of damaged epithelium and now new cells will come which have good adherence. So people are focusing on how you can, so people consider it as a wound which is there. So how you can repair this wound. So as I said, that's the reason people, this IL-22 is one. There are other mechanisms people are looking at, but so far I haven't seen something which can magically repair repair the epithelium. Yeah.
Otherwise, we can paste the gel directly, physically we can go inside.
And place, but that is also possible. But the epithelium itself, when it is damaged, it produces a number of inflammatory molecules.
It's keep on making then.
It can keep on making till it dies. Definitely putting a gel will seal, but it is not an easy thing to go into the intestine and find the place where it is and then to put a patch there so that it seals that area, right? It's skin, you see that happening much easy. It's much more easier to do than the alimentary canal. So these are molecules which are produced by inflammasomes. So usually they will give. So all these are inflammatory response, and these are just molecules which will give different kind of inflammatory response. So this is TSLP, IL-33, IL-25 are considered Th2 kind of inflammatory responses, and these are usually initiated when you have a warm infection, like you have parasitic warm infection. Then these are more important. These are more important when you have other bacterial and normal infections. And here also, both Th2 and this kind of an immune response is basically for fighting worms. If you remember when I showed you in the beginning, immune cells, eosinophils are important in fighting worms. So that's why those become activated. So this is more or less what you are already what was already shown in the video, that you have both, in the T cells you have Tregs, in the B cells you have IgA secreting B cells. And this is just showing you how the antigen is transported via the M cell, which you saw in the video also. So you have M cells in between which can help transport. You can also have DCs which can sample directly. Okay. So this is showing that okay, ultimately it will lead to activation of B cells and production of this IgA. Need not worry. If the immune system is activated, ultimately the B cells will be activated and they will produce whatever antibody they are supposed to produce. So here they are showing that even this, when epithelium produces this APRIL BAFF, these factors are known that they can act directly on the B cells. You don't need the T cell help in this case. And then the B cell can become activated and secrete IgA. This is what I mentioned with IL-22 along with IL-7. See, IL-22 is important for epithelial repair, but IL-22, when it is present along with IL-17, like during an infection, then it also enhances the production of antimicrobial peptides from epithelial cells. So basically, if you see what is going to happen, some infection is coming in, epithelium is getting activated, it is immune cells are getting activated, but you want this immune response to enhance. So whatever they are producing is going to enhance the function of all the other cells, so that now they collectively can fight the infection better and better. So all the cytokines which are produced, cytokines are just means how the cells will communicate with each other, because they cannot move, epithelial cells cannot move. But if a cytokine is produced and there is a receptor on the epithelial cells, it can respond. So that's what happens. If there's more IL-17, IL-22 in the milieu, that's why I said, if it is together with IL-17, then it induces a lot of defensins, and it is shown that it enhances production of antidefensins and other peptides. IL-22 is also produced alone. In that case, it is more involved in repair and regeneration. Now there is no inflammation. IL-17 is not there. So that is what I meant. So this I already showed you that small intestine has a much less diverse commensal community, but it is vulnerable to worms and norovirus. And then there is another disease known as celiac disease, in which basically people are allergic to gluten. You know, gluten is something which is found in all kinds of flours. So they really can't have any kind of wheat or products. So, and you may have heard that people become allergic to nuts, peanuts, milk also. So these are, and it is, and you will see, now you hear more about these things, and there's a lot of interest in finding out why these people are reacting to this food, because in general, as I mentioned, our system is evolved in such a way that we don't respond to these foods, to any food which we eat as such. So people think that maybe their microbiome is different, or the suppression mechanism in these people is maybe not as as efficient. But right now nothing is known. That's an area which is relatively open. And this is simply to show that largest intestine has more microbial community, and it has different infections which are there. And ulcerative colitis and basically inflammatory bowel disease and ulcerative colitis, and these are all similar kind of disease but have different ways they affect. So this is just showing you that commensal bacteria are very good at stimulating Treg responses, and this I mentioned earlier also, that short chain fatty acids can act on antigen presenting cells to suppress inflammation. And this class of bacteria, commensal bacteria, Firmicutes, produce this. So people believe that if you enhance these commensal bacteria in your system, you know, by probiotic, you may be able to suppress inflammation. So this is only one class which has been studied. There are other things which can also do that. So similarly, this is another class which produces this, which enhances Treg pool. So basically there are many different ways where the Tregs are generated. So if we find out how the tolerance is maintained in the mucosa, then maybe you can develop a therapy for diseases like inflammatory bowel disease. So basically when there is an alteration in the gut environment because of antibiotics, diet, hygiene, pollutants, even stress, infection, if they're more prone to some kind of a genetic predisposition, something what is known as dysbiosis occurs. Basically, dysbiosis means the good bacteria go down, and there is an increase in this the invaders and pathobiological or pathological kind of bacteria. So that's why you have inflammation. So basically, it is very well now, it is kind of more or less believed that the microbiome definitely changes in case of inflammatory bowel disease syndrome. So I think I've already shown you this. I will not keep on showing you and talking to you about all these things. So this is what I've showed you, that IL-22 acts it increases the production of antimicrobial peptides. So apart from the intestine, there are other barrier systems also, right? Our lung.
If IBS patients are taking antibiotic, then also they should not get any inflammation, right?
But see, the antibiotic does not distinguish between the good bacteria or the bad bacteria. Yeah. So it is going to kill the bacteria, pathogenic, but it is also going to kill the beneficial bacteria. So that's why I'm not sure how much effective it will be. You can, because you are also disturbing the
normal
microbiota. That's why I don't think, that's why you see that long term they are not given long term antibiotic therapy in case of inflammatory bowel disease. So now if we move to the respiratory or respiratory mucosal immune system, now the difference is from the intestine is, this is also a very large area, but this is responding more to inhaled air. So now here the epithelial cells are in contact with air, and so that's why they have some special features. One is the cilia, because cilia beat and they sweep the microbes away.
So can we put this stem cell into the bowel area so that it
this epithelial cell is, even though both are epithelial cells, I told you that in this it is modified, this epithelial cells has changed in such a way that it needs to have air on one side and
that's what, ma'am. Can we engineer this like a CAR-T yesterday we have discussed, no, ma'am?
So why do you want to engineer?
Because the pathogen will not enter inside and cause inflammation.
Because of the cilia, you think?
Yeah, you said no, ma'am, sweeping.
Yeah, but villi are also there in the intestine, right?
But this is more specialized, right?
But then it is specialized, but more for the infections which come in via the air. So it throws, yeah.
Absorbed food in intestine, it is necessary that the food should be absorbed and identified in such a way that it will absorb the food as well as secrete the digestive enzymes. This ciliary epithelium is not made for that. So if you put the stem cells of these epithelial cells in the digestive system, you won't be able to digest the food and absorb the food, which is the function of that area.
Thank you. So they have these club cell, I think these are not goblet cells which secrete mucus. Then don't worry about dust cells, but their macrophages are in the, you have a lot of alveolar macrophages in the lung, and they are tolerogenic because as it is a mucosal site, you need to have slightly suppressed immune system. And these are basically the lymph nodes associated with the nasal, with the lung mucosa. So this is just showing you how it is organized. The alveolar macrophages, bronchial lymph nodes, and something is coming in. And for this also, the reason I haven't put too many things is I have a lot of I have a video for this also, which I think simply shows you it should be connected, right? Yeah. If it gives a problem, then we will see. It will come. It's a set of videos from the nature group for mucosa, and all of them were given some award or something. So I personally feel that they are YouTube कहाँ चला आ रहा है लोड हो रहा है
The lungs are where the body draws in vital oxygen and exchanges it for carbon dioxide.
हाँ रिवर्स कर दे उसको धक्का लग जाता है
But every breath we take is accompanied by potentially harmful microbes, as well as particulates which can damage delicate lung
थोड़ा सा ही ऐसा हुआ है
tissue. To counter these The lungs are where the body draws in vital oxygen and exchanges it for carbon dioxide. But every breath we take is accompanied by potentially harmful microbes, as well as particulates which can damage delicate lung tissue. To counter these threats, the lungs are enriched in immune cells that repulse invaders and repair injury. But this potent arsenal comes at a heavy cost. Overreactions or inappropriate responses result in diseases such as asthma. The lungs are spongy organs composed of increasingly finer tubules. First comes the trachea and the bronchi. These are then divided into the bronchioles, which are wrapped in a muscular sheath. At the ends of the bronchioles are microscopic air-filled sacs called alveoli. In the case of asthma, most of the harmful immunological events occur within the bronchi and bronchioles. The bronchial wall is made up of an epithelium covered in tiny hair-like structures called cilia, an interstitium, and muscle cells. Goblet cells scattered along the epithelium discharge a thin layer of mucus into the bronchiole lumen. The mucus is continually swept back up through the lungs by the action of the cilia. This system is known as the mucus escalator and is important for removing inhaled particles trapped within the mucus. Embedded throughout the bronchiole wall are immune cells such as dendritic cells which sample antigens from the lumen, mast cells which are packed with granules, tiny packages rich in histamine, and innate lymphoid cells, or ILCs. Individuals with asthma need to have been previously sensitized to a specific environmental antigen. The most common is found in the feces of house dust mites. But symptoms can also be worsened by respiratory virus infections, smoke, or pollutants. When inhaled antigens contact the epithelium, they trigger the release of two chemicals, IL-25 and TSLP, which stimulate nearby dendritic cells and innate lymphoid cells, those ILCs. The dendritic cells leave the epithelium for the lymph nodes, where they activate T cells and initiate an adaptive immune response. Antigen can also pass through the epithelium, especially in conjunction with damaging particles such as those found within tobacco smoke or diesel fumes. Some of this antigen can reach the mast cells housed in smooth muscle tissue. The mast cells of asthmatic individuals are unusual. They have more of an immune molecule called IgE stuck to their surface. The attached IgE then binds the relevant antigen, in this case from house dust mites, in a highly specific manner. When an antigen gets bound to two IgE molecules, it signals the mast cell to degranulate and spill out the contents of the granules. The granules' innards then exert their effects on surrounding cells and tissues. In asthma, the granule contents cause smooth muscle contraction and mucosal edema, leading to narrowing of the bronchioles. Many anti-allergy drugs target this key process of degranulation. Back to those T cells in the lymph nodes. Once activated, T cells enter the interstitium and team up with ILCs to escalate the asthmatic response. They secrete chemical signals that recruit other immune cells, most critical of these being eosinophils. These cells play central roles in almost all allergic diseases. Eosinophils squeeze through the epithelium into the lumen and begin releasing their own inflammatory signals, including IL-5, IL-13, and platelet activating factor. Together, these act on goblet cells, causing them to ramp up production of mucus in a process known as goblet hyperplasia. By now, the victim is in the midst of an asthma attack. As the attack proceeds, more and more eosinophils are recruited into the lumen and the mucus layer progressively thickens. The collective effects of smooth muscle contraction and mucus overproduction restrict airflow and result in the classic symptoms of asthma, including difficulty breathing, coughing, and wheezing. Multiple attacks can, over time, lead to fibrosis and permanent injury of the lung.
So this was showing you how basically asthma occurs, because the basic homeostasis mechanisms in lung and intestine, the epithelium still secretes those retinoic acid and other factors which I was telling you to keep the immune system in check. And here, because you have so many of these alveolar macrophages and the whole, so that's why the alveolar, instead of the dendritic cells, it is more the alveolar macrophages in the lung which help control the inflammation, because their numbers are so much, and they are distributed much more in all areas, that in fact in our lab we had cultured dendritic cells with epithelial cells, and we had also cultured macrophages, alveolar-like macrophages with dendritic cells, with epithelial cells. And what we found was that the epithelium makes the macrophages more tolerogenic, like it makes them immunosuppressed, but it enhances expression of toll-like receptors and other pathogen sensing molecules on dendritic cells. So initially we were very surprised, because it was generally believed that the whole of the immune system cells are suppressed. But if you think about it, it is fine, because the number of dendritic cells in general is lower, and if you have tolerance on one hand, you need to have some kind of a compensatory mechanism, in the sense which should, so that you don't get infections. So this increased expression of toll-like receptors and other molecules on dendritic cells will enhance immune surveillance, because now they have more receptors which can survey for any kind of a pathogenic bacteria, so that you don't get infections all the time, because as I said, the macrophages will take a little longer to get activated. So maybe it's the way we have evolved is we always have checks and balances in the immune system, you will see. It's only when that balance changes that it results in some disease or the other. So here you saw an example of asthma, but okay. I think this has gone back to, I'll just check. Okay. So what are the infection which all of us know for lung? Most common is the influenza, and influenza is a single-stranded RNA virus. So there are two major pathogen recognition receptors which are involved in recognition of influenza. One is the TLR7, and the other is the RIG1. And in it's not only the TLRs and the RIG1 which are involved, but other proteins of influenza can also bind NLRs and activate the inflammasome. And NK cells can kill infected cells which display this HA molecule of influenza. So what this is simply showing you is that the B cells also produce antibodies. So there is all the cells are doing whatever they can to fight any kind of infection. Here is an example of influenza, but a similar thing would occur in other infections also. And in case of allergy and asthma, it is, as you saw, that why we get this, because these dust mites, even pollens, trigger release of molecules like TSLP and others from the epithelial cells, which change the environment in such a way that now cells will produce, T cells will produce more IL-4, IL-5, and as you can see, it acts on mast cells and then eosinophils. And you have heard of the term eosinophilia, right? In many cases, many people, and that is basically what it was showing. When you have a lot of eosinophils in your airway, it causes all those problems and causes difficulty in breathing. So the reason these two things are mentioned, because these are the most common of the problems associated with the lung: allergy, asthma on one hand, and respiratory infections on the other. So the other reason why people are very much also interested, apart from fighting infections, also are very much interested in understanding how the lung mucosal immunity functions, is because it is a way where you can deliver local, vaccine to the lungs. And flu mist has already been developed and used also. Flu mist is basically you deliver an aerosol of the vaccine, so you don't have to inject vaccine. There is a lot of interest in developing needle-free vaccines because the advantage are manifold. One is you do not require a very trained personnel to give the vaccine. So, and the other thing which people are also very much interested in developing vaccines which are more stable, because right now the major problem in compliance is if you take it to rural areas and other places, you need to maintain a cold chain so that the vaccine.
Last month also I saw on paper BCG, they are trying through, like what you said, nasal. Instead of giving BCG injection to kids, they will cry and they'll.
So what people think is that for infections which are for the lung, if you can deliver via this nasal route, by flu mist or whatever spray or whatever it is, it induces the kind of immunity which is required, because now you're realizing that the immunity in the lung is a little different than what you would find in the systemic. So that's why there's more focus on this kind of delivery systems. So, and you all know that you need to have a new flu vaccine every year because the flu vaccine, the flu virus is such a virus that it changes its envelope proteins. It mutates the envelope proteins very fast. So the part which is recognized by the immune system, this envelopes, is different all the time. And so based on previous infections, drift and everything, scientists decide, okay, this year maybe we will have a combination of these three kind of flu viruses. So the vaccine is made. It sometimes is very successful, sometimes it is not. So, but there's no better mechanism right now. So there's a lot of work which is going on where people are trying to make a universal influenza vaccine, which does not need to be changed every year. So they're looking for molecules which do not change with this mutation. And can you make a vaccine using these kind of molecules is what is being tried. Some of the structural proteins are known not to change. So those are the ones which are. So now we move to the next mucosal immune system organ, and that is the skin. So skin has, the top layer is epithelial, but it is known as keratinocytes because it contains this protein keratin, a large amount of this. And this keratin is very important because it forms a very thick it's a very impermeable kind of protein. So because you need protection on the skin so that things don't penetrate easily. So it is like a layer of dead cells which form this hard kind of shell, which is not as much of a shell as what you see in crustacean, but it is an invisible kind of shell which is there. Also, so top layers are usually dead, as I mentioned, and then you have the dendritic cells in the skin. They are known as Langerhans cells because they contain special kind of granules known as langerine. So that's why the name Langerhans cells has come. And I had mentioned TRM. These are tissue resident memory cells, and this is the reason people think that the vaccine. So I forgot to mention this, but when you are giving vaccines via local route, you will generate local memory also, right? So that's the reason that is also one of the reasons why people think that local delivery, wherever the infection is supposed to occur, you may get better kind of a immune protective response. And most of the immune cells, so epidermis is the top layer followed by the dermis. Usually the epidermis is kind of dead cells and only the lower layer is the living cell. Dermis is where you will find all the immune cells and everything, other cells, and that is what is mentioned here. In fact, you will laugh, but I have a video for this also. So, so you can see this. Give me 2-3 minutes and it will come.
This dead cell basically protects us, no, ma'am.
Yes.
But when we get dandruff, that is also dead cells only. But
it's not in the skin, right? It's in the so dandruff is it's still shedding of the. See this? So it's the same people who have made these videos.
The skin is the body's primary barrier against physical insults and microbial pathogens. It represents a unique environment in which immune cells interact with skin cells to maintain tissue homeostasis and induce immune responses. The skin is composed of epidermis, dermis, and a subcutaneous fatty region. Commensal bacteria, fungi, and viruses living on the skin have beneficial effects in the protection against pathogens and in wound healing. The epidermis is composed of highly specialized epithelial cells known as keratinocytes. They are continuously replenished from just one layer of basal keratinocytes, which divide frequently. Dead cells called corneocytes form the outermost layer and are largely responsible for the barrier function of the skin. In the dermis, cells known as fibroblasts secrete elastin and collagen fibers that form a dense extracellular matrix. Blood capillaries irrigate the dermis, while lymph fluid is drained through lymphatic vessels to lymph nodes, specialized immune structures in which immune cells are activated after pathogen encounter. Diverse and functionally specialized immune cells populate the skin. In the epidermis, a specialized subset of dendritic cells, called Langerhans cells, sample antigen. They project dendrites upward towards the cornified epithelial layer and sample bacterial antigens such as toxins. Langerhans cells appear to be both anti-inflammatory and activatory depending on context. Dendritic cells in the dermis are highly efficient at capturing dead cells and presenting antigen, such as viruses, other intracellular pathogens, or skin-associated self-antigen, to T cells. If dendritic cells are the immune sentinels, T cells are the immune effectors. Healthy skin contains more than twice the number of T cells found in the blood. Most of them are memory T cells that have previously encountered antigen and can be rapidly reactivated. T cells in the epidermis are mostly CD8 T cells, a subset that becomes cytotoxic and kill target cells upon activation. They're long-term residents in the epidermis, mostly disconnected from the circulation. T cells in the dermis are mostly helper CD4 T cells, which have a more modulatory role in the immune response. A variety of other immune cells, such as natural killer cells, eosinophils, and mast cells, are present in the dermis and might be involved in allergic reactions in the skin. Dendritic cells and keratinocytes sense tissue damage, such as wounds or cold sore lesions that occur when latent herpes virus reactivates, and they do that through evolutionarily conserved receptors that recognize pathogen-derived molecular patterns or host-derived molecules that are exposed by cell death, such as DNA. Keratinocytes produce antimicrobial peptides, which can kill bacteria directly, inflammatory mediators, such as interleukin-1 or IL-1, which activate dendritic cells, and chemokines, which recruit neutrophils, macrophages, and T cells. Activated dendritic cells migrate to the lymph nodes, where they present antigen from the site of infection to naive T cells, priming them to activate and differentiate into effector T cells. Activated T cells return to the skin and kill infected keratinocytes to control viral infection or secrete signals that recruit additional immune effector cells. Following viral clearance, memory CD8 T cells persist in the epidermis to provide immunity for future encounters with the same virus. Immune responses can become dysregulated and cause skin disorders such as psoriasis or atopic dermatitis. Psoriasis is a lifelong inflammatory skin disease characterized by scaly reddish plaques. A combination of environmental and genetic factors confers susceptibility to the disease. Physical injury or inflammation can trigger the formation of an acute lesion. The antigenic trigger is unknown, but current models propose that stressed keratinocytes might release self-DNA, which in complex with an antimicrobial peptide activates dermal plasmacytoid dendritic cells to secrete high amounts of the antiviral mediator interferon. Together with pro-inflammatory IL-1 alpha released by stressed keratinocytes, interferon activates dermal dendritic cells to promote T cell differentiation. The earliest recognizable change in the affected skin is the accumulation of T cells and dendritic cells around blood vessels in the dermis. An overt lesion occurs when CD8 T cells, dendritic cells, and neutrophils infiltrate the epidermis. Specialized subsets of T cells secrete soluble mediators like interferon gamma and IL-17, which stimulate the proliferation of keratinocytes, and this produces a marked thickening of the epidermis. Signals from the proliferating keratinocytes act as chemoattractants for infiltrating neutrophils. Crosstalk between immune cells, keratinocytes, and dermal cells thus contributes to tissue remodeling and amplification of this dysregulated immune response. Without treatment, acute psoriatic lesions become chronic lesions. Genetic studies have identified psoriasis-associated susceptibility genes, some of them linking Th17 cells, the subset of T cells that produce IL-17, to psoriasis pathogenesis. Thus, immune cells in the skin exert important roles in maintaining the barrier function against pathogens, but can also become activated by self-antigen or harmless antigen to cause autoimmunity or allergies.
So basically, I think more or less, as you see that each time there is some change in the homeostatic balance. One is infection. Infection can occur, usually it is taken care of and everything goes back to normal. The problems are more chronic infections or chronic diseases where the balance remains changed, then it leads to more problems. So you saw like the inflammatory bowel disease, you saw psoriasis here, and in the lung you saw asthma. These are all chronic conditions in which the system has lost its balance and it is constantly activated in one way or the other, and that is what we need to control. So now I think with the advent of immunotherapy for cancer, there is now a lot of focus on finding immunotherapies for these diseases also. And as I said, there is a major interest in how you can induce T regulatory cells. And here I have not shown you, but vitamin D on this, if you take vitamin D is also known to induce high levels of T regulatory cells. So many of these vitamins, we know that their deficiency causes what disease, but generally those are like very high level deficiency, like vitamin C deficiency leading to scurvy and all those things. Those are now not that common kind of thing. But low level deficiency of these vitamins can also cause problems. And vitamin D is, in fact, it has been shown even in people sometimes are using it in conjunction with some therapies like higher doses. And one of the other things which people have found is like vitamin D is the vitamin which is made by our skin, our body when we are exposed to sun. So in theory, we should not have deficiency here. We are exposed to sun quite a bit. But it has been found that in darker skinned people, it seems to be the synthesis of this vitamin is not as efficient. And many a times, even though we are exposed to sun, we still are deficient in vitamin D levels. So that's and in the intestine, people are trying this, at least in the mouse, I have definitely seen where people are trying to give vitamin D as a therapy in the sense that it suppresses. So vitamin D basically acts on dendritic cells to suppress, make them tolerogenic. It also acts directly on T cells to make them induce Tregs. So that's why a lot of some people are working on this kind of, because
does it have a receptor and it goes directly into the nucleus?
No, it has the vitamin D has a receptor known as vitamin D receptor. It is in the cytoplasm. It's not too much on the surface, but it goes in, because it is a lipid soluble molecule, so it does go in. And what else should I say? In fact, it has been shown that during obesity, this vitamin D levels, for some reason or the other, seem to be decreased. And there is some correlation between maybe leptin, one of the obesity-related hormones, and this. And some people even believe that if you will read the literature, that vitamin D suppresses inflammation. It can also help lose weight or help in management of weight. So, and I already told you yesterday that with another vitamin, biotin, that its deficiency is causing intestinal inflammation. So now we have two of them, and vitamin A induces production of retinoic acid. So that also is known to induce Tregs. So you can see how important all these things are for our natural health. So see, there's a lot of interest suddenly in these, how do I say, you can say it alternative medicine, or these kind of compounds which we have regularly, and we have had them over the years, right? And they haven't resulted in any major kind of pathology. And we have a history, okay, it's like passed down from generation to generation, that if you have this, it will control this, right? In our food, we already know, okay, don't eat warm food, don't eat cold food if you have this, don't eat this. The only thing which is lacking, and which seems to be happening quite a bit here, where they are focusing on validating how these things work. Because right now the science behind them is less clear. So we have a whole, like I would say, a treasure of different plants and different things which we use. The only thing which is lacking is we may not know how they work. We do know that they work, And so I've talked to a number of faculty here, and they are working on different areas, which and they're trying to find out the reason why those things are, how mechanisms, how these things are working, because then the thing becomes more acceptable in general. And maybe you can devise some kind of a drug-based, another drug based on that, maybe some modification, maybe which improves its bioavailability. The other problem is that things are cleared out very easily from our system. So I think it kind of concludes my today's lecture. And this is that there are different kind of areas in the mucosal immune system, and you need to have an interaction between the microbiome and the barrier. It can be beneficial or pathogenic, and then you need to have a balance of tolerogenic and inflammatory response to the microbiome. Okay, so if you all have any more questions, you can ask.
Ma'am, ma'am, with this celiac disease, simply but just by avoiding gluten-containing food, how can we treat that disease?
By avoiding gluten-containing food, you are preventing the irritation, basically. So it will not recur very frequently. Basically, you are removing the trigger which is going to activate. So that is why people say avoid the gluten-inducing.
Because my brother had this problem, so doctor just say that simply just avoid that food. But it can, when he eats something, just
because some of the things contain gluten which we don't even know. Because sometimes there's even obvious foods are very clear. Some other proteins which may be related to gluten can also cause problems. And in some cases, you have additives which you have no idea that they are there. So that could be the reason that.
So, ma'am, can we treat this disease by simply inducing the T-Rex cells, so they can suppress their response against gluten?
So that is what is being tried in food allergy, but they also think that it should, maybe what you should do is induce antigen specific Tregs. So specific Treg specific for maybe celiac disease, and Tregs can be antigen specific. But for most food allergies, right now I would say more work is being done on peanut based allergies, because peanut or nut based allergies. The simple reason is the anaphylactic reaction or the allergic reaction to these are very high, and they can be fatal if not treated very fast. So that's why there's more focus right now on those. I'm not saying people are not working on celiac and other disease, but I see more emphasis from the physicians based on more on the nuts and this allergy. But
ma'am, with as time goes, it just treat itself, or it just stay there?
So some allergies people have seen that you have in childhood. Some allergies go away with time, but some of them seem to remain. Nuts is one. I have not seen that celiac disease kind of disappears, but I have known patients, or I've heard of patients who over time seem to develop a little bit of tolerance to it, especially when it starts very early in the childhood. Sometimes when they become adults, they maybe because you have prevented them, you've been very careful and not exposed them. I'm not 100% sure. So it seems to vary by person to person. So there is no a universal rule.
For this kind of food allergen, what they're giving a very minute concentration. Then slowly they'll increase. Then they're making it resistant.
So that you develop slowly tolerance to it. But that is mostly that it is also, so these ones, there are two ways they are doing it. Either you can inject small amounts here, and other is sublingual. Below the tongue also it's being given because it's non-invasive. But mostly I would say right now it's been I have not seen it too much for celiac disease. As I said, I see it being tried for this peanut
Peanuts.
allergy. I think what they're thinking is, if you can control such a high anaphylactic reaction by this, if you can make it work, then maybe you can use it for other, treating other food allergies.
Ma'am, if it remain untreated for a long time, can it lead to cancer?
If there is constant inflammation, it can lead to many problems. It may lead to cancer because inflammation is one of the trigger, right? Can also lead to inflammatory bowel disease for that matter, but only if it is not in control. And there is no hard and fast rule that okay, you have this, so you are going to get that. So, yeah.
Thank you, ma'am.
Thank you. No problem. So that concludes the immunology session. So first of all, I do want to say a few words that I'm very appreciative of the audience who have taken the time and really listened to everything very carefully and asked different questions. It has been very nice talking to you all, and I'm really thankful to the GIAN program, but especially to Dr. Wadegawkar. He is the one who really made me aware of this kind of a program, and he did most of the work. And here also, he and his wife have really taken care of me in um, all different ways, and I've really made them run around for many different things. And I also appreciate all the faculty which have interacted with me, and they've told me what kind of research they do. So it's very interesting to hear what is going on in this biotechnology department here. So, as I said, it has been a wonderful experience for me coming here. And I find also that students are very, very interested, and they ask very relevant questions even when I interact outside. So it's been a very, very learning and very nice experience for me. And thank you all for it. Oh, you.
Thank you. I request you all to move towards the lawn. We will have a group photograph. As soon as you are ready, Honorable Vice Chancellor will join us.
Okay. अच्छा पानी बरसने की चांसेस है हाँ मैम 7 दिन से वही चल रही है वो ना बारिश 3 बजे बारिश अच्छा उसका टाइम है कल जाते
hello, 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 hello. Hello, hello, hello.
हा तर चालू आहे पण
Hello.
आवाज येईल इकडे capture घेतली ना.
Hello, hello.
त्याच्यात आहे ते तिकडे ते जमला.
इथे चार्जेबल आहे का याचं?
Hello, 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 hello.
गुड मॉर्निंग। मैं उस देश की नागरिक हूँ जहाँ छत्रपति शिवाजी महाराज और सम्राट अशोक जैसा राजा था। मैं उस देश की नागरिक हूँ जहाँ छत्रपति शिवाजी महाराज और सम्राट अशोक जैसा राजा था। मैं उस देश की नागरिक हूँ जहाँ हर 50 मील पर रूप, पहनावा, मजहब बदल जाता था। मैं उस देश की नागरिक हूँ जहाँ डाल डाल पर सोने की चिड़िया को पाया जाता था। मैं उस देश की नागरिक हूँ जिसे अपने भूगोल से पहले इस ब्रह्मांड का भूगोल समझ आता था। A very fanatic morning to respected honorable Vice Chancellor sir, Dr. Sabkal sir, the most Dr. Anshu ma'am, and the most eminent personality of this department, Professor Dr. Wadegaonkar sir. I welcome you all the dignitaries on the behalf of Department of Biotechnology, Sant Gadgi Baba Amravati University, Amravati. Welcome you also. As we all know that tomorrow develops when we prepare for it today. To develop a budding scientist, Ministry of Human Resource Development comes with an international level global initiative of academic networks, that is GIAN, in higher education. With respect to that program, from last few days, our department has organized a workshop, and it was very successful. As this is the last day, I have got the opportunity to host this program as a end of ceremony. On that occasion, now I request Dr. Respected Vice Chancellor sir, to welcome our visiting faculty, Dr. Anshu ma'am, for with a little gift. Please, sir. Thank you so much, sir. As it was a program we have conducted, and all we have the participants from all the universities, and we all the research scholars, students, and all the expertise were this program. Now, as the end of a program, I request anybody volunteer as a feedback. Uh, Please, from audience. Thank you, sir.
Respected dignitaries and faculty members and my dear colleagues and friends. Actually, this is the first time I have come from Odisha to this state of Maharashtra. Actually, and before um, coming here, actually, I was contacting Sir Vadagaon sir, and he was very cooperative. And finally, I got registered and I came here. So after coming here, before coming here, I was actually searching about the profile of the faculty member, and then I got that she's having a lot of experience in immunology, and I am also doing the same work in. But I am a veterinarian, so I am working in bovines, and especially my PhD work was this. Um, Regarding the C-type lectin receptor. So that motivated me, and I thought that it will be quite helpful to me in my future research career. So I came here, and the session was very interactive and full of young students, and the course was designed in such a manner that it will motivate the students for taking up their future career, and also we, as we are already in a job, and we have been I think we have been recharged by the our faculty, and the course was very interactive, and she was answering the queries of the students very nicely, and it was very interactive. And the as far as the hospitality is concerned, it is quite very nice, and faculties and the students are very cooperative, and so thank you all.
Any more feedback from audience? Please. Good morning all over here. We are the students from Amity University, and we are very privileged. We feel very privileged and thankful to Wadigaonkar sir, Anshu ma'am, and other members who actually took care of us. We never felt left out. We felt very taken care of, just like we feel in our own university. And the course was very educative and very informative. We had our own seminar. We felt like we were in our own home place. Thank you so much. Thank you. So from last few days, we came through the topics like innate immunity and adaptive immunity. We also studied the antigen processing and presenting dendritic cells, and also immunotherapeutic approaches using dendritic cells for treatment of various diseases, including cancer. Dr. Anshu ma'am has explained very well. As we are the students of MSc, till now we was only introduced to the basic immunology, but after attending this workshop, we came to know how we can instead of introducing various chemicals or allelopathic drugs in our body, why not to modify our immune system in such a way that the disease can be cured at our own body level. And what happens in general, when we go to the doctors, they give us some same drug to all the patients. By attending this workshop, we came to know the body cells differs in each and every person, and depending on that type of body cells, now the scientists are designing various drugs for us. Thank you, ma'am, for this introductory speech. Now, I request Vice Chancellor, sir, please enlighten us. Please, sir.
Respected dignitaries on the dais and their students from various states and our university also. On Sunday, this GIAN started, and from this Sunday I am out of Amravati. Yesterday night I came here, and today you are going. This is concluding day. And after 10 minutes also I have meeting, the Incubation and Innovation Center. I have less time. But I want to discuss with you because this is also first time for the university and maybe for you also. And so many things they introduced by the MHRD and UGC regarding the quality in education. That is one of the GAN also. I want to share with you regarding the experience, what you have learned and what is your experience in this workshop. But it is unfortunate part for me. I cannot do presently. One thing you realize in this workshop, so many things are coming in higher education, new things, and maybe in some year, the entire system and so many things will be changed in higher education. Now, totally evaluation of the examination and our education is depend upon the 3 hours examination only, and on the basis of curriculum, whatever be prescribed by the university and your board only. Just you see the depth of your subject and scenario that subject also. I feel in my subject I belong to commerce faculty. Only maybe more than 1% again, 2 or 3% is part of the ours entire scenario of our subject. Now only 3 to 4% I learn and appear the examination, get the results. And then I say, yes, I am success man in my subject. When after completion we go practically in the field of our topic, then I realize my knowledge, the very much less the knowledge whatever I got in my student life. That is the major problem, and that is the reason we cannot get the properly output from the education system. That's why this such type of workshop is very important. This GAN you got maybe I could not interact with you. That's why I'm not confident about this. So many things, those who are not part of your curriculum and syllabus, the madam here only because of GAN. You can take the knowledge from the madam, who is the excellent personality. Only because of GAN she is here, and you can get the knowledge. We must go beyond our syllabus curriculum for get the knowledge, and this is the GAN is very helpful to our students. You don't attach to get the knowledge other than syllabus and curriculum with GAN only. This will be habit. What are the new things are coming in my subject? That should be a bit, yes, I will get the new things and develop myself. That is the motto of the GAN, and I wish you all students and congratulate also, you are fortunate. This is the first batch in our university and you can join here, and this is the congratulate part our university also. You are here. Again, thank you and all the best.
Thank you so much, sir. We are enough fortunate that today we are having Professor Dr. B.S. Sabkar sir from Department of Chemical Technology. Now I request Sabkar sir, please enlighten us. Please, sir.
Very good morning to all of you. We are brother, chemical technology and biotechnology, these two departments. We started together. I was just passing through it. Maregaonkar sir stopped me. He said, I have been, of course, following what Honorable Madam was delivering for last 5 days on YouTube. And he said join. And meanwhile, Vice Chancellor also came, and then it happened that then बोलते ना बिना बुलाए हुए then on the dais. Nevertheless, but it's always I'm very happy. As a Vice Chancellor in Nagpur University, when we also tried to start this GIAN program initiated by MHRD, and as you know that my background is chemical engineering, so more of chemical engineering professors were contacted, and the very main purpose of this program is, the name itself says, what is that? GIAN. So you have to add GIAN through global initiative to your existing GIAN. And that is, once you realize that what is the magnitude of the gap between recent development in particular field and what you are practicing, what knowledge you have, master's level, PhD level, and what is now needed to upgrade you with respect to recent developments, recent advances in terms of knowledge, in terms of practice, in terms of processes, in terms of instrument design, and in terms of technology. Today's world is global digital university. As you know that online programs are available, advanced programs are available, NPTEL or you name it, National Digital Library is available to you. Similarly, these global programs are also available to you. But since there is necessary to have the more interactive session, that's why MHRD has invited a lot of international experts in a selected field. Though this was a basic immunology program, but it is, of course, you can analyze that what advanced knowledge from this basic program in your existing knowledge you can get, and you have to map the incremental rise in your level of attainment. Understand, level of attainment in your knowledge is required to be mapped in the form of whether you have improved the capability of designing, capability of what you call creating some sort of new, now if it is biotechnology, some sort of new process, technology. And as you know that whole globe today is looking at United Nations various, what you call last 1 week, it's happening, whether it is Global Economic Forum, whether it is Global Climatic Change, or whether it is bilateral or multilateral, all people all over the world, the top bosses are in US today, interacting and trying to identify the area of cooperation. And if you see really the agreement made in various with India or between the other countries are dedicated to upliftment of the health of the human, which has been highly impacted because of the climate change, because of diversity, biodiversity impact, and everyone is trying to now get the commitment about the control of climate change. And hence these technologies are coming to the help now to the human being, whether it is yesterday it was declared that India can be the next what you call health hub, health treatment hub in our country, where a lot of facilities are coming up, and then they will be based on not only experts. It's a global now. I'm sure that if Madam US में रहती थी, so she would have attended Modi's lecture of Howdy Modi and then talk about what are the areas of cooperation between the universities. And as you know that a lot of changes is taking place globally to share the expertise, to share the facilities, and also share the what you call knowledge in order to improve the basically human life. And that is why we are talking, and along with of course the climate and the global issues. As you know that GIAN has given you opportunity to keep yourself updated, stay current with the knowledge, and opportunity to make furtherance in your existing knowledge and see that you have now recent and what you call advanced level of knowledge, which will be helpful to you, not only for your future career, but also in the pursuit of your study and research. I hope that you got a lot of important inputs, and the difference between the methodology of explaining the concept and proof of concept. I'm sure that these two aspects have been also emphasized during this workshop. I was recently in Singapore's Nanyang Technological University, where a big class is given, only the concept is explained and the proof of concept is solved in tutorial by way of designing the new problems and solving the problem by sharing the solutions. The problems are to be solved by sharing the solutions, and the solutions are to be analyzed with respect to its applicability and viability. By this way, you must also gain the knowledge of a new trend in learning process as well, and also making your learning resources optimally utilized. I'm sure that I'm very thankful to Dr. Wadegaonkar sir. Of course, the university is also benefited, students are benefited, and everyone, faculty, and this activity with all within the framework of the university, and all faculty members of biotechnology department and the students and volunteers of this department has made it possible to transform the treasure of knowledge through madam to you, and I congratulate all of you and Dr. Wadegaonkar and all of his team, and we look forward more such programs in future, so that we have n number of opportunities on the select topic, and it is between the collaborative, it is between the collaboration that the foreign faculty should agree on the topic, the content, and also the timeline, so that the students can be benefited. With this, I once again congratulate the department and also all of you, and I hope that this is the beginning of a new way of learning and enriching your knowledge. Thank you very much.
Thank you so much, sir. As sir has explained, the magnitude of gap should be fulfilled somewhere. After attending this workshop, we are really, our knowledge has increased, and the magnitude of gap will really fulfill. And now I request Dr. Anshu ma'am, please enlighten us. Please, ma'am.
So, as I already mentioned, it has been a pleasure interacting with you all, especially it is good to see how our youth is asking all the right questions, thinking in the right direction, and there's also enthusiasm to develop new things, and that has been very interesting for me to see. And one of the things, people think that I have come from US, I find the students here no different. They are equally eager, equally enthusiastic, and willing to do whatever is required. And the other thing which I really liked was coming to this Amravati University, which I mentioned earlier also. It's so nice to see such a nice green campus, and which is very rare to see in most places. The hospitality here has been extraordinary, and my every need has been taken care of immediately. And all the students have been highly interactive. So it has been a very, very learning and a very pleasurable experience for me while coming here. And I'm definitely very, very thankful to Dr. Wadegaonkar for introducing me to this kind of a program, because to teach you all so many different topics in immunology, I also had to read a lot. So I feel that I also benefited a lot because of this program. Thank you.
Thank you so much, ma'am. Now it's time to have a guidance from a person who always keeps struggling for students and and the man behind the camera who is actually the backbone of this successful program. Now I request Dr. Vadegaonkar sir, please enlighten us. Please, sir.
Thank you. Dr. Anshu, Professor Sapkar, and all dear participants. Professor Sapkar is my elder brother, I will say. We joined in 1995 with one chair, one table. Yeah. What should I say? We started like that, and this is a department which today I am heading the department. Professor Sapkar is established and heading the department since 1995, and I got almost all type of support, help, and that too without any formality. Means even in 1995 and today 2019, if I needs anything from his department, any of my student can walk in and get that thing there. From that type of support, I always got from him. So unfortunately, on first day, it was a Sunday. He was also not available. Professor Chandekar was also not available, and I was also not keeping well the time. So I could not approach to everyone in time. But we started this course. Sir, we started when we got the A grade from the NAC in 2016, and in 2017 MHRD opened the avenue for us, and from that day, the day which I got the responsibility to coordinate as a coordinator for the university, we started searching the resource persons. We started looking for different programs, and we submitted 4 programs to the MHRD. It was a tough time for us. We were struggling to request them to review our proposals. See, we are the new, means in their list, this university was a new university. Maximum programs goes to the IITs, and in that to a place without airport is asking for the GIAN program. But I'm really pleased to share with you that we submitted the 4 programs, and really I'm grateful to the MHRD and reviewers. They sanctioned 3 programs, and this was the first program. You can understand our problems. We contacted Dr. Anshu in 2017, then just trying to update. Yes, ho jayega ma'am. 2017 passed, 2018 passed, 2019 May, June, I think. Finally, we got all the sanctions. Then problems start that she should get this right time, the leave. She has to manage her own schedules. I know that she is going to come back in November again, but she is rushing back to the USA because she is on the panel of reviewers for the NIH. I know that the whole next month is going to be tough for her, but even then she has spared time and she has visited our university, and I hope she has enjoyed. We really enjoyed her company. About the participants, the first participant who contacted me is Dr. Khan, who walked in my room with asking, can we also join the course? Then that time I realized, yes, we have to remain open, and I have to personally at least respond to each and every one who calls me. Immediately within few days I got calls from the Raipur. The whole group is sitting here from MIT University, and then there was a call from Andhra. Sir, आपको form मिला? Have you received our letter? Have you ever booked our rooms? I really enjoy. I was traveling that time, but really I was enjoying their comments. And I will tell you, I had not arranged anything for anyone that time. But you know, I have very much confidence on my students in this department, my colleagues, and especially for my university. And then we somehow managed the guest house, HREC guest house. As you know that it was written there that you had to pay separately for the accommodation. But I'm really grateful to our Vice Chancellor also and our whole administration who waived all the fee, those charges. I'm sure that you all have enjoyed this course. Honorable Pro Vice Chancellor Dean and Dean Science and Faculty both have promised us that they will modify the rules and they will allow us to add this course as an extra credit to the students. So we have taken already one examination, correct? I will float some next week. I will float the another question, one set of examination on the same platform, and I will request you that you should attempt that also. So we will have the evaluation also, which is essential for giving the credit to the students. So we will be ready from our side. As I said, the 3 programs have been sanctioned to this university, and for the common topic which is coming up in the month of December, that is on the evidence-based medicine. So I will again request you that if you find that topic suitable to you, please start enrolling that also. And for that, Dr. Anita Patil, professor in biotechnology, will be the coordinator. Of course, I am looking after the university coordinator that time, but she will be the course coordinator, and you will be in direct contact with her. So I welcome you all of you for that course also. Of course, I have to keep the space for the newcomers also. So with this, I wish you all the best, and after some time the certificates will be available, lunch will be served outside, and with this I officially declare that our course is over. I officially thank MHRD and Government of India for financing this course, giving us an opportunity and giving the opportunity to our students to interact and learn from the faculty like Dr. Anshu. Thank you very much.
Thank you so much, sir. As this was a very successful program, for to making this program a successful one, the various peoples have helped a lot. I thankful all the helpful hands who make this program directly or indirectly a successful one. And with the permission of HOD, Dr. Vadigaonkar sir, I declare that the program is over. Thank you.